Dear Nobel laureates, excellencies, ladies and gentlemen, good morning and welcome to today's symposium in honor of the 2023 Nobel Prize laureates. For those of you watching live stream, I'm delighted that you are joining us online for today's conversation. Today we have the privilege to honor 5 Nobel laureates who have contributed to a better world. Professor Moungi Bawendi and Professor Louis Brus, Bawendi and Louis Brus shared the Nobel Prize in Chemistry with Dr. Alexei Yekimov for the discovery and synthesis of quantum dots. I hope we will all understand a little bit more about quantum dots later today. Professor Katalin, Karikó and Professor Weissman shared the Nobel Prize in Physiology and Medicine for their discoveries concerning nucleoside base modifications that enable the development of effective mRNA vaccines against COVID-19. And all of us understand what that meant for saving millions and millions of lives in the COVID pandemic. Professor Claudia Goldin was awarded the Swedish Central Bank, Sveriges Riksbank's Prize in Economic Science in memory of Alfred Nobel for having advanced our understanding of women's labor market outcome. A warm congratulations to all of you and a big applaud. And make sure to have fun in Sweden, okay? You will have an exciting week ahead of you, and don't forget to have fun. Before I hand it over to our moderator, I would like to acknowledge our sponsors who have made today's event possible. AstraZeneca, thank you for supporting science and innovation. And finally, a big thank to the National Academy of Sciences under the leadership of Marcia McNutt. On behalf of my colleagues at the Embassy, thank you very much, Dr. McNutt and your team, for your outstanding effort and for co-hosting today's symposium. Now it's my great pleasure to welcome today's moderator, Dr. Maria Brogren, Science Counselor at the Swedish Embassy.
Thank you, Ambassador Ahlin. Nobel Prize laureates, excellencies, ladies and gentlemen. Welcome to the 2023 Nobel Prize Symposium here at the House of Sweden. As mentioned, my name is Maria Brogren and I am the Science Counselor here at the Embassy of Sweden. And today we celebrate the scientific discoveries of this year's US-based Nobel Prize laureates. It is a tradition here at the Embassy of Sweden that we organize this scientific symposium and invite this year's laureates. And we are spoiled in that sense that there are usually a couple of US-based laureates. And this year, there are no less than 7 US-based Nobel Prize laureates, and 5 of them are here today, and we are truly grateful for your presence. And I'm not an expert in any of the fields that we will dive into today. We have the laureates of the Chemistry Prize, the Medicine Prize, and the Prize in Economic Sciences here. But I'm very eager to learn from the laureates, and hopefully you will also learn something. At least that's my goal with my interviews with the laureates later on. And after my interview with the laureates, you will also get a chance to ask questions, the questions I might have forgotten and didn't realize that were interesting. So you will get your chance, and I will make sure that happens. And before we dive into the details of the different prizes, I would like to introduce 2 keynote speakers to set the scene to why we are here today and to provide a background to this important prize. First, I'd like to welcome Dr. Marcia McNutt, President of the US National Academy of Sciences. The Embassy is grateful for the close collaboration that we have with the National Academy, which Ambassador Ahlin already mentioned. And we have this collaboration around promoting science and international research collaboration. And we're also very proud to co-host the event with you. And I'm sure that Marcia McNutt is well known to everyone here in the audience, but I would nevertheless like to take the opportunity to highlight a few items on her extensive CV. Firstly, Marcia McNutt has a PhD in Earth Sciences, and she is trained in underwater demolition by the Navy SEALs. I'm very impressed by that. She was also the director of the US Geological Survey, and now she was the first woman to be elected to head the National Academy of Sciences. So, warm welcome, Marcia McNutt. The mic is yours.
So, good morning, everyone, and I really appreciate the generosity of the House of Sweden in allowing me a few remarks today. And if I could have my first slide. Oh, I've got a clicker. Oh, here we go. Yay. Whoop. So let me start by asking the question, why is this gathering happening here today in the United States and not in Germany? Okay, so let me go through some of the reasons. The US presently leads the world in the Nobel tally. I'm just going to confine this discussion today to the science prizes, not the literature and peace prize. And if we look at just the science prizes, the US leads the world in the Nobel tally with 368 prizes in the sciences. Now, that wasn't always the case. Prior to the end of World War II, Germany led that tally with 39 Nobels, as compared with second place, which was the UK, and the US was somewhere down the list with 17. So why did this change happen after World War II, such that the US started gaining on these major science supporting nations? Well, let me put up a couple possible factors that we can test. First of all, the end of World War II, many of you may remember, or at least have heard, of the document called Science, the Endless Frontier, that was written by Vannevar Bush. He was the science advisor to the president, and in this document, he made the case for investment in science and technology as an investment that would increase the productivity of the nation and improve quality of life. Investment in science didn't need to just be a wartime effort. And the nation was very receptive to this message because they recognized how important science had been in winning the war. And they bought into the idea that there would be other benefits. As a result of that report, there was the founding of the National Science Foundation just a few years later that devoted its funding to basic research. And of course, these prizes that we are recognizing today are all in the category of basic research, regardless of how soon they ended up creating actual measurable, real products that improved our quality of life. And then the third possible reason for this change is the welcoming environment that the US created for the best and brightest from all over the world to come here and study and ply their trade in science. Now, I'm going to put this caveat. It's very difficult to determine cause and effect for many reasons, not the least of which is because Nobels are a lagging indicator of quality. You have to put the input in over here before you see that output of the Nobel Prize honoring it. And the time lag can vary. The average lag for chemistry in the past decade was 30 years between the critical report or publication that was then honored decades later. But both mRNA and high-temperature superconductivity were, so in the life sciences and in physical sciences, were recognized within a few years of those discoveries. So the lag can be less, and it is field dependent. So here's just a plot of the total US Nobel Prizes. And this plot goes from the beginning of the awarding of the prize up to the present day. And you can see that there's a suggestion in this curve that the publication of The Endless Frontier and the founding of the NSF, if you add a decade or two lag, does seem to correlate with the increase in US prizes. I find it's even more compelling when you break it down by field. Here's physics, and here's The Endless Frontier and NSF. It's a pretty clear correlation that a national imperative to fund basic research paid off, and it paid off mightily in terms of this Nobel tally. If we look at Nobel Prizes in chemistry, again, you have to put a longer lag in chemistry, but there does seem to be a correlation. And finally, here's physiology and medicine. Again, a suggestion of a correlation. And then in economics, Nobel Prizes in economics, those did not start until around the 1970s. And so, of course, there's not a clear jump with the Endless Frontier and the NSF. But as I was compiling this data, something really stood out to me. And that is the number of prizes in these science fields that went to immigrants, first-generation immigrants, people who were born overseas and came to this country. You can see that in all of these prizes, it's a substantial fraction. And if we took away those immigrants, the US leadership doesn't look so startling anymore. So I thought, what about other nations? Are they also seeing an impact on immigration in terms of their ability to be competitive for Nobels? So here's a plot I put together that's very simple. What I did was I ranked the top 10 countries in terms of their Nobels. And then I calculated how many of those Nobels were for first-generation immigrants. And you see down in the lower left, USA, number 1 in terms of Nobels, number 1 in terms of the number that went to immigrants. You see on the right hand, Russia, number 10 in terms of its Nobel Prizes, and number 10 in terms of the number that went to immigrants. And then you can see that you could draw pretty much a straight line through that with a few exceptions. Germany and Japan stand out as having more Nobels with lower percentage of immigrants receiving them. I think that's an indication of the value of losing World War II. Because these two nations were on the losing side, they were forbidden from investing in military R&D. And therefore, I think it's a reasonable suggestion that they put more effort into basic research, non-military R&D, and it paid off in terms of the Nobel tally. We've got Sweden and Austria are tied in this diagram, but you see they're not far off. And Canada has a high percentage of immigrants, but not as high on the Nobel tally. And I think that's because of the fact that Canada is another New World country, and so was valuable. Now, so what my thought is on this is that the reason why the US got the leadership in the Nobel tally is the same reason that if your soccer team only recruits from your neighborhood, you're not going to be as competitive as if your soccer team recruits from the entire world. And the US had the advantage of being able to recruit from the entire world and therefore built a science enterprise that was unparalleled. But I worry about the future. I worry about the future because the US federal investment in basic research has been falling as a percent of GDP at the same time that other nations like China have been increasing their investment in basic research. Other nations are also providing more resources to retain their best and brightest. It used to be that a scientist, a would-be aspiring scientist from many nations felt that the US was the place they had to go to get their research supported. That's not the case anymore. Many of these countries from which we have recruited are now offering very good packages for their own scientists to stay or to return. And finally, I'm very worried about anti-immigrant sentiments here in the US. I think we have to realize the contributions of immigrants to US quality of life, our national security, and everything else. And so I hope that the messages you will take away from this are that we need to once again become the most welcoming nation for the best and brightest scientists from wherever, and we have to support basic research. So thank you.
Thank you, Marcia. And now I have the honor to introduce Dr. Anna Sjöström Douagi, Vice President of Strategic Initiatives at the Nobel Foundation, who will give an introduction to the great donor Alfred Nobel and the reason why we are here today. Anna Sjöström Douagi has a PhD in immunology from Karolinska Institute in Stockholm, Sweden, and she is a member of the Royal Academy of Sciences in Sweden. Welcome, Anna.
Thank you so much, Maria, and thank you, Marcia, for a wonderful presentation about the Nobel Prize here in the US. I'm so happy to be here today in this wonderful day here in DC, where the sun is shining in this beautiful house of Sweden. Thank you for hosting this seminar. And first of all, I would, of course, like to say big and warm congratulations to all of you laureates from me and my colleagues, and we are very much looking forward to seeing you in Stockholm in just some weeks. So, and actually relating to the speech by Marcia McNutt, on December 9, we will have a seminar on the future of migration together with Nobel laureates and other experts, and it will be broadcasted online, so it's just go into our webpage, nobelprize.org, and you register. So, talking about Stockholm, some in the room are soon going there, and we are very, very happy to welcome you. This is the City Hall of Stockholm. It's enlightened by light art. It's a light festival in Stockholm in December in the honor of you and the Nobel Prize. And there is spots on different places in the cities that you can walk around, so it's enlightening the whole city. And there, that is also the building where the Nobel banquet is being held. And it feels very important at this time to highlight the ideas of Alfred Nobel, and also his idea of the importance of international community, the respect for knowledge, and also the belief in humankind's ability to bring about change, as you all have been proven. And you are all carriers of these ideas and beliefs now, and you will be a great source of inspiration for us and for the future generations. So I'm just taking a quick introduction of Alfred Nobel and why there is a prize before coming to your discussions and presentation about your prize-awarded work. So who was Alfred Nobel and why is there a prize? Alfred Nobel, there is not so many portraits of him. Here is one, actually painted after his death. He was born in 1833 in Stockholm. He was one of 8 children. His father was a quite famous inventor. And early on, they moved to Russia, St. Petersburg, to try the Russian market there. And he grew up in this very cosmopolitan atmosphere. He studied both at home, but also abroad. He studied chemistry in Paris, he studied technology here in the US, and he spoke 6 languages and lived in many countries. And in Paris, he met the person who discovered nitroglycerin. And he got really fascinated by this and this explosive matter, how to do that in controlled forms. And that was something he would spend many years of his life trying to do. And as you all know, he eventually succeeded, and that led to the invention of dynamite, which he patented in 1867. But that was not the only patent. He was continuously working, and he had, in the end, 355 patents. and he grew this enormous fortune. He had businesses all over the world, and he was a very international person who believed in globalization, also in a time where the nationalism was quite strong. So, and beyond that, he also had a big passion for literature. So he was reading a lot, he loved his library, and he actually tried to write some poems and novels with people saying with various results. Unfortunately, he lived a very lonely life, was completely dedicated to work, so he died alone, which he feared the most, but that actually happened. He fell in love with Bertha von Suttner, who for a while worked as his secretary. Unfortunately, she left to marry another man. So he wasn't that lucky in love, but he was very romantic. Bertha von Suttner and Alfred Nobel, their friendship lasted for life, and they were corresponding heavily. She was a great peace activist, and they corresponded about peace and arms. And is it good with a lot of arms or not a lot of arms to create peace? She believed to take away all arms. Alfred Nobel thought about if we have enough of arms, it will make peace in the world. She is believed to be a great source of inspiration for the Nobel Peace Prize, a prize that she herself was awarded in 1905. The last couple of years of Alfred Nobel's life, he wrote several wills. The third version was signed in 1895. People believe that that was not supposed to be the last version, but it actually became the last version because 1 year later, in 1896, on December 10, he died alone in Paris, or no, in Italy, where he lived at that time. So he left this enormous fortune. He had brothers and kids, and to their great disappointment, he decided to leave his fortune to you. So you are the heirs of the Alfred Nobel, and he wanted to create a prize that will ensure the continuous development of intellectual and cultural progress in the world. And the prize would go to persons who had conferred the greatest benefit to humankind. And that was irrespective of nationality or gender for that matter. And that was very controversial at this time. The Swedish king was not that happy because you didn't need to be Swedish to get this prize. And also the fact that the Peace Prize were awarded in Norway. So it was quite political, it was many tensions. But the first prize could then eventually be handed out in 1901. The first physics prize went to the discovery of X-ray, which was something that Alfred Nobel himself was extremely fascinated about. So, as you are well aware of, there are some prize awarding institutions that are deciding on the prizes. It is the Royal Swedish Academy of Sciences up to the left, deciding on the prizes in chemistry, physics, and economic sciences. There is the Nobel Assembly at the Karolinska Institutet, which decides on the medicine or physiology. There is the Swedish Academy deciding on the Literature Prize, and there is the Peace Institute in Oslo deciding on the Nobel Peace Prize. And these institutions, they get nominations. They work for that for a year. They are already starting to work with the next years. And then it's actually decided on the same day that the prizes are announced, and you get an early call here in the US, and then it's just before we have taken the decision. So it's a very happy day. So the strongest symbol of the Nobel Prize is, of course, the medal, which you will soon receive. But there are three insignias that you will receive when receiving the Nobel Prize. So it's the medal in 18-karat gold, but there is also a handmade, unique diploma made foremost by a Swedish and Norwegian artist. But then there is a small amount of money as well included. Right now it's approximately $1 million US, and it can be shared by no more than 3 persons. So to date, the prize, we got some beautiful statistics here from the US, but to date, the prize has been awarded to 621 times and to 1,000 person, and you were the 1,000th person who got the Nobel Prize. And also 27 organizations. But since some people actually got the prize, not only 2 times, but 3 times, there are 965 individuals who got the prize and 27 organizations. And we are very happy to see an increase also in female laureates in the last couple of years. So I just need to show one thing. did Alfred Nobel succeed in his wish to create this prize for the benefit of humankind? So I asked AI to make a presentation for me about the Nobel Prize, and this is what I got. So it recognized that the Nobel Prize was about laureates and your achievements, but it also said that it has a very prestigious recognition, it has global impact on the society, it creates a legacy of excellence, inspiring future generations, encourage innovation, creativity, and groundbreaking discoveries. I think Alfred would have been very happy about this result. So on December 10, 127 years after Alfred Nobel died, there will be a ceremony in the Stockholm Concert Hall where the Nobel Prize will be handed over to you by the Majesty the King. On the podium, you have the representation of the different branches of the Nobel organization. You have the donor, Alfred Nobel, in the middle. To the right, there is the prize awarding institutions and the Nobel Foundation. To the left, you have the Nobel laureates, and then you have the royal family who is handing over the prize. In Stockholm, when you come, you will visit a quite small Nobel Prize Museum, which was established 20 years ago. It was a traveling exhibition, but it was permanented. Since then, we have seen an increasing demand on you and the Nobel Prize to work to inspire both youth and the society in general. So therefore, we will build a brand new Nobel Center in Stockholm ready in 2031. That will be a house where your work and discoveries will be the basis for activities all year around. And it's really with the aim to create a platform for new ideas and discussions. So you are now a part of the history of Alfred Nobel, and we welcome you to the Nobel family. And together we keep Alfred Nobel's legacy alive, that science, literature, and peace work has made the world a better place and will continue doing so. And hopefully the challenges of today will be solved by the laureates of tomorrow. Thank you so much.
Thank you, Anna, for sharing these insights into the life of the great donor Alfred Nobel, and also some insights about the ceremony in Stockholm. That will be in 4 weeks' time. Now I had the honor to introduce the first Nobel Prize laureates. We will start with the Chemistry Prize, as that is because we go by the order that the prizes are mentioned in Alfred Nobel's will. And he starts with the Physics Prize, and then it's the Chemistry Prize. So please welcome Louis, Brus, and Moungi Bawendi up on the stage.
Okay.
So we have Louis, Brus here. Louis Brus and Moungi Bawendi share their Nobel Prize in Chemistry with Alexei Yekimov for the discovery and synthesis of quantum dots. And first, I will introduce the laureates briefly, and then we will discuss their discoveries. Louis Brus was born in Cleveland, Ohio, and entered Rice University in Houston with a scholarship from the Navy. He graduated at Rice with a bachelor's degree in chemistry, physics, and mathematics, and moved to Columbia University for his doctoral research. After his PhD in chemical physics, Brus served as a scientist at the Naval Research Laboratory here in Washington, before joining Bell Laboratories, where he did the experimental and theoretical work that led to the discovery of size-dependent property of quantum dots, and where he also met and collaborated with Moungi Bawendi. In 1996, Brus left Bell Labs to go back to Columbia University, and today he is the Samuel Latham Mitchell Professor Emeritus at Columbia. Moungi Bawendi was born in Paris, France, but the family migrated, as we heard, migrated to the US when he was a child and lived in Indiana, as Bawendi's father worked at Purdue University. Bawendi received his bachelor and master from Harvard and earned his PhD in chemistry from University of Chicago. During his PhD, Bawendi participated in a summer program at Bell Labs, where Louis Brus introduced him to the research on quantum dots. And after receiving his PhD, Bawendi went to work with Brus at Bell Labs as a postdoc. After that, Bawendi joined MIT in 1990 and became a professor there in 1996. Today, he is the Lester Wolf Professor of Chemistry at MIT. So, Professor Brus and Professor Bawendi, you are awarded the Nobel Prize in Chemistry for discovery and synthesis of quantum dots. And we, of course, like to know more about this. Professor Brus, can you explain to us what quantum dots are and why you became interested in them?
Well, a quantum dot is a very small rock, basically. That's the one way to describe it, with maybe crystalline material with maybe 10 atoms across in diameter from one side to the other, made and characterized using the methods of chemistry. And what's interesting about them is they have some properties which are molecular in nature and some properties which are solid state in nature, bulk crystal type. And so there's a size transformation. You take a very small little cluster and let it grow larger by adding atom by atom, and eventually it looks a little bit like the small rock, and then eventually picks up properties of solid state materials, either metallic or semiconducting. So it's this size regime which is intermediate between the interests of chemistry and physics and the methods of chemistry and the methods of physics.
And how did you get interested in this field?
Well, it was an accidental discovery. Like many things in nature, I was making semiconductor colloids to study the surface organic photochemistry when they were excited by light, absorbing a photon, and then there'd be a molecule on the surface, and that molecule might be oxidized or reduced. And I had spectroscopic methods for studying this, and I made colloids that sometimes showed a larger band gap than the bulk band gap of the larger particles. And so I stumbled into this regime where the properties began to change as you went to smaller size because of this fundamental transformation, because of quantum effects. And we realized pretty quickly that this was interesting basic research, because we were exploring a new regime, new materials, making new materials, a new regime, but it was basic research that not immediately but ultimately would be important in the electronics industry, because transistors are made out of small pieces of semiconductor, and you can have 100 million transistors on one chip, and the thing that makes them better and cheaper with time is the fact that they're getting individual transistors are smaller and smaller. So eventually the engineering of semiconductor chips would enter this regime where the properties of the silicon being used to make the transistors were no longer the same properties as bulk silicon. You'd have to understand the properties of how things changed as size went smaller in order to change the engineering. So it was basic research, not immediately useful in the next year or so, in the next 10 years, but we had confidence it was a good project.
And when you first observed this phenomenon, did you know the size of the particles, or could you control them, or how did you know that it was size dependent?
Yeah, so the main method was the electron microscope. You can make a colloid and then take the particles out and put them on a substrate and look at them with the electron microscope. They're far too small to see with an optical microscope. You know, the particle is 50 angstroms in diameter. The wavelength of light is 5,000 angstroms, so you can't see them. They're, as far as light is concerned, they're molecules. They behave just like molecules. Looked at them with the electron microscope. We could see that, well, there were a lot of problems. There's distribution of sizes, distribution of shapes, you know, and distribution of surface chemistry on the little particles, and extremely heterogeneous sample. And so, besides working on simple theory, we spent lots of time trying to improve the, in an Edisonian way mostly, the synthesis, if that's the right word, method of making the particles to make them more monodisperse and more highly crystalline, you know, and control the surface chemistry. I used to say that, you know, for every hour I spent doing spectroscopy, laser spectroscopy is my original interest, but I spent 5 hours doing synthesis for every hour, you know, because we worked on the, we have to work on the thing that needs basic improvement.
And Professor Bawendi, when did you get interested in quantum dots?
Well, my background had been laser spectroscopy of very small gas phase molecules and also statistical mechanical problems in theory. But when I arrived at Bell Labs as a graduate student and I discovered the world of this intermediate regime between condensed matter physics and molecular chemistry, I just fell in love with it. It was a brand new area of science. And in any brand new area of science, every time you ask a question, it's a new question. And every time you make a discovery, it's a new discovery. And that's just phenomenally exciting when you're a young graduate student.
And speaking about, I mean, new questions and so, but you also shared the prize with Professor Dr. Jekimov. Were you aware of his work at the time, or did you sort of go this path in parallel?
Well, I'll let you
Okay, so
do that one.
we were not aware. I mean, Alexei was publishing in Russian behind the Iron Curtain. Now, he was not allowed to travel to the West and give talks in the West, and his work was just not known. And he clearly saw these effects maybe a year or two before I did, in a different medium, in high temperature molten glass rather than in a room temperature colloid. I was not aware of it, and I worked out the theory independently. And then after about a year, I finally found through literature search one of his papers that had been translated. And so I wrote into the Soviet Union to Alexei and to his collaborator Sasha Efros and told them I was working on this also, and we tried to make friends. He couldn't come out, you know, I was not going into his institution, so I didn't meet him for another 5 years or 8 years. We only made close contact at the time of Gorbachev began to lead the Soviet Union and perestroika. So that's how it worked out.
Thank you. Professor Bawendi, you sort of, I think that you sort of set your mind to try and control the particles more, the size of them. Can you explain how you did that?
Well, as Lewis's discovery was somewhat accidental, mine also was a lot of trial and error. Lewis, working with his collaborators at Bell Labs, Mike Steigerwald and Paul Visados, had developed a method for making the particles, which was largely room temperature, where they figured out how to basically wrap each particle in a set of molecules, organic molecules, as if you were to wrap a particle in a plastic bag to isolate them. And they had discovered that, you know, you could make the crystallinity a little bit better by heating them up. And I came there and, you know, tried to heat them up at higher temperatures with different molecules, different solvents, and sort of accidentally encountered one particular kind of mixture that worked really well. And because my interest was really in the measurement process, you know, once I had that as a postdoc, I did a lot of measurements on it. Then I went to MIT, and we could make, at the time, we could make one size pretty well, and it was reasonably well crystalline, you know, and sometimes it worked and sometimes it didn't work, but it worked well enough that you could we really begin to study the physics. So at MIT, I had this program of understanding the physics, because that's what my love was, the curiosity of this transition from the molecules to the solid state and how it worked. But the first thing we needed was material to study. And so when I tried with my students to reproduce what I had done at Bell Labs, it worked some of the times. At MIT, it just didn't work. Different latitude, but I'm not quite sure. So we had to reinvent the process. We went to higher temperatures. We ended up making a manufacturing process, or a process for making the particles, which was basically a one-step process. It had been simplified. We looked at the ideas from the 1950s physical chemistry of nucleation and growth and applied them to the growth of these particles. And so we were able to create much tighter distributions. Because of the higher temperatures, the crystallinity was also much better. And there's an interesting parallel between the way that Ekimov grew his particles in molten glass and the way that we ended up growing them in high temperature solutions. Both of them are high temperature, and both of them eventually used the ideas of separating nucleation and then growing on a uniform distribution of seeds slowly, so that all the seeds grow at the same rate to get to the nice particle. Then you can arrest this at any time to get your particle sizes.
So what was the material? What did the particles consist of?
So at the time, cadmium selenide was the material that we all studied. And part of the reason for that is because it all happens indivisible. So you can see it with your eye. And so you can make this solution, and then the small particles let all visible light go through, so the solutions look yellowish or even transparent, basically colorless. And as the particles grow, they begin to absorb more and more of the light spectrum. So by the time they're very large, almost bulk-like, the solution has a very deep, deep red color. And so you can just see with your own eyes the growth of the quantum mechanical process. So it's basically seeing quantum mechanics with your eyes, which is really amazing when you're a student, because you learn about quantum mechanics, and then suddenly you can just see it,
So
and
you mean it changes colors?
As you're growing the particles, right? So if you were to use something like cadmium sulfide or something with a smaller band gap, everything would happen either in the UV or the infrared, and then you wouldn't be able to see it. But the ability to see it with your own eyes means that the feedback is very rapid. You try something, you know immediately that it's not working, and so you can adjust on the fly.
So is this material also used in solar cells nowadays?
The research that people are doing using for solar tends to use a smaller band gap material, for instance, lead sulfide, that has the band gap that's much more suited to the solar spectrum. But cadmium selenide has become the hydrogen atom of these particles, the hydrogen atom of nanoscience in a way, because we know so much today about it. So if we have an idea about how things change, it's usually the first material we apply it to do the experiments on, to understand things.
Because you know it so well, so you can
Yes.
you have a baseline.
Exactly.
Yeah. So did you continue to collaborate after you were not in the same department anymore? And
We did, yes.
We actually worked together on the luminescence of single quantum dots. This was maybe 2 or 3 years after Moungi left to go to MIT, and on and off, there was always something that we might do together. But basically, separately, I went on to work on, when Moungi left, I went on to work on silicon quantum dots rather than cadmium selenide. Very different bulk semiconductor, and much more relevant, obviously, to the computer industry. And but the bonding in silicon is so strong that can't be made by these liquid methods. And so I worked on an high temperature aerosol method, and we worked through the characterization and understanding of that. while Moungi was investing the years to perfect the synthesis of the cadmium selenide, as he described, you know.
But this, the silicon process, is that done in vacuum, or how is that done?
No, it's done in nitrogen or argon gas, you know, running through an oven, 800 centigrade, and then passivating the surface, first making good little silicon crystallite quantum dot, and then passivating it with introducing oxygen to build up maybe 10 angstroms of silicon dioxide on the surface. Then converting that back into a colloid. So I would bubble that through ethylene glycol solution, you know, and then we would have a colloid, and then we could use the colloidal methods that we were working with for cadmium selenide, you know, liquid chromatography, separation of size, you know, and size selective precipitation, and those methods that Moungi had worked out with his students to further improve the size distribution.
So you could control the process to get more homogeneous size, but you could also separate them, to sort them,
yes,
size-wise.
That's right.
Once you have particles that are well formed and crystalline, then you can separate them out because they're dispersed in a solution. But the bigger particles have stronger forces that attract them to each other than the smaller particles. So if you change the solution properties ever so slightly, then you can get the biggest particles to come out as a solid, leaving behind the smaller particles. You wouldn't be able to do that if the particles weren't all well formed. So they really need to be, you know, little spheres that are all coated in exactly the same way to be able to do that.
I mean, that's basically the value of this chemistry approach, the liquid approach, is that, you know, once you get a rough understanding of what's going on, then you can make particles that are designed. You know, for example, you can make core shell particles that start with cadmium selenide and then change the reagents and add reagents that will make zinc sulfide on the surface rather than cadmium selenide. Then that will change the physical properties. And then you can take them out of the solution at the end of this synthetic process and then use them for something else. You know, so that's, you can see why we think it's a branch of chemistry, yeah.
We're actually out of time now for our discussion here, but there will be time for questions from the audience in the end. So hopefully we can sort of talk a little bit more about applications, for example. But please stay seated. Okay. So now we will invite the next laureates. We will move on to the prize in medicine or physiology. And I would like to welcome the laureates, Katalin, Karikó, and Weissman, up on the stage. The Nobel Prize in Physiology or Medicine is awarded jointly to Katalin, Karikó, and Weissman for their discoveries concerning nucleoside base modifications that enable the development of effective mRNA vaccines against COVID-19. And I will also start with a short presentation of the laureates and then some questions for them. Katalin Karikó was born in Hungary and obtained her Bachelor in Science in Biology and her PhD in Biochemistry from the University of Szeged. She continued with postdoc research at the Institute of Biochemistry at the Biological Research Center in Hungary. In 1985, Karikó was offered a research position at Temple University in Pennsylvania and left Hungary for the United States. Also another immigrant who got a prize. After Temple, she worked at the Uniformed Services University of the Health Services before joining Perelman School of Medicine at the University of Pennsylvania in 1989 to work on messenger RNA. Drew Weissman was born in Lexington, Massachusetts, and received his bachelor and master's degree from Brandeis University, where he majored in biochemistry and enzymology. He performed his graduate work in immunology and microbiology at Boston University, where he received his MD and PhD. After that, Weissman did a residency at Beth Elizabeth Deaconess Medical Center, followed by a fellowship at the National Institute of Health, under the supervision of Anthony Fauci, whom I'm sure that all of you know. In 1997, Weissman moved to the University of Pennsylvania to start a laboratory to study RNA and innate immune system biology. And that's where he met Katalin Karikó at the famous copy machine. And they began to collaborate to move the mRNA technology forward. And I would like to start our discussion there when they met. So I'm very happy to have you here, Professor Karikó and Professor Weissman. And I would like to ask you, Professor Karikó, can you briefly explain to me and the audience what mRNA is and what function it has in our bodies?
Yes, so the messenger RNA, not what we discovered with Brus, it was discovered in 1961, and this messenger RNA is present in all of our cells, and it carries the information from the DNA to the protein factory, which is the ribosome, and short-lived and information. This is a middleman, you know, kind of what we could sometimes say, yeah, true.
So it is actually the messenger.
The messenger,
Yes.
and then it takes the message, and then the protein is made, and then it degrades.
So Professor Weissman, what is the difference between mRNA vaccines and the traditional types of vaccine that we had before?
So most vaccines that we get when we're kids and even as adults are based on a few different platforms. There are live viruses that are attenuated. The measles, mumps, rubella are live viruses. Some are protein subunits. Tetanus toxoid is the classic. Some are inactivated viruses where they grow the pathogenic virus and then inactivate it. Influenza is the classic. The RNA and DNA and adenovirus are known as genetic vaccines. They don't have the proteins. They have the information that codes for the proteins. So with the mRNA vaccine, the RNA codes for the spike protein from coronavirus. It causes our cells to make that protein, which is recognized as foreign, and our body makes an immune response against it.
So it's not the old types of vaccine you sort of poke at the body to make this, but with the mRNA you get the message. So it's another way of doing it.
Yep, exactly.
Yes. And also in the prize motivation, there's a mentioning of nucleoside base modification. Professor Karikó, what is that, and why were they needed in this case?
So when we started to work together with Drew, we discovered that this mRNA, which I was making, is very inflammatory. And we were curious to understand why. And we did a series of experiments where we discovered that not all of the RNA is equal. Transfer RNA was not inflammatory, and that gave us the idea that maybe, knowing that the tRNA has a lot of nucleoside modification, we thought that maybe if we modify the nucleoside in our mRNA, maybe we can make non-inflammatory molecule. And that's how we get into this question that, okay, now that we have to make a RNA which has modified nucleoside, but nobody did those things, and we didn't know whether it is doable. And so that's, we tried out whatever was commercially available. We tried to incorporate already modified nucleoside triphosphate to incorporate to the RNA, and turned out that when uridine was modified in that mRNA, it was not inflammatory. And we zeroed in the pseudouridine because we get more protein, and the RNA was not inflammatory.
And in practice, this modification, how is it done? Is it chemistry or?
I mean, in our body, all of the RNA we make from the 4 basic nucleotides, and then different enzymes changes it. but we couldn't do this way because the enzymes were not known and we couldn't modify it. So we purchased nucleoside triphosphate, which already was modified, and we, tried to cross our fingers that the polymerase will incorporate. And so that's how we made it.
So it was the combination of the mRNA and this substance that sort of...
I have to say that naturally we have in our body the most abundant nucleoside, actually pseudouridine. And when we did these experiments that incorporated into the messenger RNA this pseudouridine, it was not known that actually naturally in our mRNA also we have pseudouridine. Actually, 10 years later they discovered that we also have it. So that's how we incorporated, and that's how we tested, and we found that it was superior.
Yeah. And when you tested the tests, how were they performed to test that it was not inflammatory? Perhaps Dr. Weissman, were you involved in that?
So when I left the NIH, I studied a cell known as a dendritic cell, and it was a newly identified immune cell. And what its job is it circulates all throughout the body, and it identifies foreign, it identifies pathogens. It picks those pathogens up, brings it to lymphoid organs, and stimulates an immune response. So it's the central player in all infections, and importantly in vaccines. So when I got to Penn, I wanted to use them as vaccine platforms. With mRNA, the first experiment we did, Katie made RNA, I added to the dendritic cells, and went back a few hours later and looked at them, and they were incredibly activated. And when we talked about this, it made no sense because our cells are full of RNA. So why should a natural product be so inflammatory? And we continue to use those dendritic cells as a way of measuring RNA activation.
And so then you changed and you used the same sort of platform or method of evaluating.
Yes, we evaluated the inflammation and we also did the translation. So we could see that it translates well, and we could see that it was not inflammatory. So in our research, compared to, we both of us were needed. The RNA and the inflammation. If I made the RNA actually 10 years, almost worked 10 years on mRNA research, I did not notice this inflammatory, and it was critical that Drew has this cell and he tested, and so both of us work was needed.
Yeah, perfect. And then I'm curious, we all know that this, it's also mentioned in the prize, how this directly relates to the vaccines that we have used now during the pandemic. How was your research translated into that? Were you involved in these companies, or?
So we actually started making RNA LNP vaccines 5 or 6 years before the first publication ever came out. The first major publication was in 2017 for the Zika pandemic, where we made a vaccine where a single immunization of mice or rhesus macaques protected them from Zika. So the vaccine was done. It was made, the platform was made. Moderna started clinical trials for influenza soon after that. When COVID hit, it was an established platform. And all it took was plugging the sequence from the coronavirus into the RNA. And within 2 months, patients were receiving the vaccine. So the platform was made years beforehand and was in patients years beforehand. I often talk to lay audiences and they say, oh, I'm nervous about the vaccine because it was invented in 10 months. And that's just as far from the truth as possible.
So are there the next step for vaccines? What are they? What vaccines are you looking to use this platform for?
So there are many companies making vaccines right now. In clinical trials are vaccines for rabies, for CMV, for influenza, for HIV, and many more are entering clinical trials for norovirus, for C. diff, for malaria, for tuberculosis. The potential of RNA and vaccines is enormous. Vaccines for autoimmune diseases and food and environmental allergens are also being developed. So the potential is just unbelievable.
Actually, I was not wanted to make a vaccine. It was
No.
always Drew wanted
Yes.
to make the vaccine.
Yes.
I
So
was,
that's why
I
we
was
can-
worried about the inflammation because I want to make it therapeutic.
Yes.
And that's what actually 10 years ago when I went to BioNTech, I was also there responsible for the RNA-based protein production. So antibodies, what we did based on mRNA, but we always collaborated, and Drew collaborated with BioNTech, and we worked together even I was in Germany. But when I came, I always visited you, isn't it?
So which diseases have you targeted?
So, you know, I work cardiology, I work in the University of Pennsylvania and worked in neurosurgery, so that always whatever was the topic there. So I try to develop therapy for a stroke patient, and then when I went to BioNTech, that did mRNA coding for antibodies or cytokines, and those were all oncology program, but not vaccines. So that's what I was in charge.
So what can, if mRNA is used in another way as a therapeutic, how does that function in the body? Is it the same principle, or what does it do?
So that if the messenger RNA codes for, we did test it for like erythropoietin, this which makes red blood cells to differentiate. And so when you deliver the mRNA code for this product, and that has biological effect, and then red blood cells number will increase, and so that's already. I was always thinking a protein which was already present in the human body. I just not enough or not the right place, and then I thought that if we deliver the mRNA, then we can increase the level, and it is transient. And if something is helpful, you can reapply, or it's not good, then you stop using it.
And is this used today?
The erythropoietin, not yet, but it will be, hopefully.
Yeah. Because I mean, as I understand it, it's then that you program the cells to behave in the way that they need to. I mean, if they need to change something, if there is a deficiency in the body, you can.
Yes, there are also programs where rare diseases treated, so that mRNA coding for a protein which the patient doesn't have sufficient amount of enzymes, for example, it is clinical trial is ongoing.
And then I'm curious, how in practice is it still given as a shot, or how do you introduce the mRNA where it needs to be?
Yeah, it's mostly still injection.
Yeah. So it works the same way as a vaccine, but it does something else to
not necessarily, not in the intramuscular. Some of them intravenously is delivered, or, for example, the program that I participated at BioNTech, it was intratumoral injection of the mRNA coding for different cytokines.
Interesting. I'm really hopeful for that, because there are so many diseases that we need more effective therapeutics. So, actually, I also, I mean, talking about therapeutics in general and vaccines, there's also a matter of cost. So, for example, Drew, I know that you are involved in making vaccines more available in low-income countries. Can you tell us a bit more about that work?
Sure. So I've worked on equity even before I was in Tony's lab, and have worked on it my entire career. 10 years ago, I started working with a group in Bangkok, Thailand, where I introduced them to RNA LNP vaccines before anybody knew what they were. And they became very interested. We established an institute in Chulalongkorn University, and they've got a half a dozen researchers working on a variety of different vaccines. When COVID hit, we met with the Princess of Thailand and the government, and they concluded that any vaccine that was made in the West would take years before Southeast Asia, Sub-Saharan Africa, South America would ever see that vaccine. They weren't willing to wait. So the princess and the government put up money to build a GMP manufacturing site. GMP manufacture is the grade of drug that can be given to humans. So I worked with them to build a GMP site in Thailand. After that, I started working with the WHO. We've built 15 GMP sites across South America, Sub-Saharan Africa, Eastern Europe, Southeast Asia. The most fantastic is we're building one in the Ukraine right now, in the middle of a war. I met with the people doing this in Cape Town a few months ago. They say it's incredibly difficult, as you can imagine, but that they're building it, it's up and running, and they'll soon be able to make GMP drug.
Perfect. Thank you, both of you, so much. Really learned a lot. Thank you. Yes, and now let's move on to our last laureate, last but definitely not least. Now it's my privilege to introduce Professor Claudia Goldin up on the stage. She is the winner of the Sveriges Riksbank Prize in Economic Sciences in memory of Alfred Nobel 2023. Welcome. Claudia Goldin is awarded the Prize in Economic Sciences for having advanced our understanding of women's labor market outcomes. Claudia Goldin was born in New York City and graduated from Bronx High School of Science. She intended to get a degree in microbiology, but found economics instead, and the multidisciplinary approach that became her calling. She received her bachelor degree in economics from Cornell University and a PhD in economics from the University of Chicago. After her PhD, Goldin taught at University of Wisconsin, at Princeton, and at University of Pennsylvania, before joining the economics department at Harvard, where she was the first woman to be offered tenure in that department. And she was also the first woman to be offered tenures in other economics departments. So we have a real pioneer here. We have actually two pioneers now on the stage. Few of them, I mean, as we heard, that women are increased in the Nobel laureate community. And now Claudia Goldin is the Henry Lee Professor of Economics at Harvard.
Oh, thanks very much.
So, Dr. Goldin, your research covers a wide range of topics, including the female labor force, gender gap in earnings, and how it relates to different sort of aspects of society, technology, education, and immigration, again. And most of your research interprets the present through the lens of the past. And can you tell us how you became interested in this field, in studying women's participation in the labor market?
So I'm an economic historian, and I work on a very, very large range of issues in economic history. And we work, all of us work because we're curious. I mean, that's what drives us. We read something, we have a question in our heads, and we start working on it. And at some point, I was working on 19th century families and the family economy, and suddenly I realized that everyone was writing about and exploring what the children were doing, what the male heads of households were doing. But there was very little about the mother, the wife, the adult woman. And the reason was that the sources were very difficult to find, so she was left out. And yet, it was pretty clear across the long sweep of history that suddenly this person was going to be a large fraction of the labor force, but where did that come from? Where did that happen? And so then I set out to write that story.
And then when we come back to the price and gender gap, so what do we mean by gender gap? Is there a definition?
Well, the definition would just be in some mathematical manner the difference in the fraction of women who are in this occupation versus men, or do it in some other manner, or the difference in earnings between two groups. And so we know that there are these differences. Exactly how we measure them gives us different information. There are ways that they're measured so that they're simple. Everyone likes a single number. So today you might say it's 83 cents on the dollar, and in 1960 it was 59 cents on the dollar. So that looks like it has increased a lot. But that is only one single number. And in fact, if you ask what happens to the gender gap for a woman versus a man across their life cycle, we'll see that it actually starts out being the gap is very small. That is, this ratio is about 1, and then it expands over time. And seeing that makes us ask a somewhat different question.
So, what would you ask? Would you ask why is this gap expanding at certain times in the woman's life, or?
Right. So one would want to know exactly that. Does it expand more when she starts a family, when she's married, after she graduates from college or high school or whatever? We want to follow this to understand why it expands, and then to understand the reasons why it does. And of course, I study this long period in history, and in certain periods the answer is going to be different than in other periods. There was a time, some time ago, when women's education was much less than men's. In fact, today, in every single OECD country, every single one, women attend college to a greater extent than men. That was not always the case. And in fact, in the United States, that crossover point was way back in 1980. We often hear, oh, women are graduating from college at higher rates today. Well, that's been going on for over 40 years.
I saw that you, in one of your books, you have this U-shaped curve describing women's participation in the labor market in the US during the last 200 years or something like that. Can you explain what does this curve show?
So this U-shape is replicated in many different countries and in a cross-section across countries. And the reason is that historically, when the home and work, let's say in the family business or family farm, were one, it was very easy for an individual who had child care responsibilities or other care responsibilities to do what we think of as market production close to home. So we see in countries in which markets are not as well developed that women actually have very high labor force participation rates. As economies develop and as markets expand and as the wages of individuals rise, if they leave the home and go to large-scale enterprises, then women in some sense sort of retreat back into the home. So that's the downward portion of the U. And then for various reasons, we get the upward portion as wages rise relative to home-produced goods, as bread is baked in factories and not in the home, for example, as electricity enters the home and therefore reduces the price of home-produced goods. And that's sort of the historical story. But this is played out across countries all over the world.
Yeah, so it's, I mean, to narrow it down, is it about, I mean, where you are located, if you can provide for your children, you can take care of your children close to where you work, then it's... Did this change during the pandemic, when more people, both women and men, started to work from home? Can you see some results already from what happened?
So we're still sort of too close to that to get a perfect read on it, but it certainly does seem as if right now a large fraction, I know the data for the US, although even the data for the US isn't totally clear, but almost all studies show that today about 30% of paid days work are done at home, paid days work. And there are, in certain studies, differences between men and women, and in other studies, they're about the same. So we're just too close to it to know. But we know that something big has happened. I mean, the pandemic was horrible and ugly, and if not for the work of the people sitting to my left, we would probably be less out of it than we are, or it would have taken us longer to get out of it. We thank them tremendously. Now that we're out of it, we see changes that we could always have had. We always had these technologies sitting around to work from home, to do conferences with people across the globe without going there. And these changes have really affected how we work, and we're not quite certain exactly what they're going to do, but they certainly seem as if they're affecting women a bit more than men, because if you don't have to go to Tokyo to sign a contract, it means that you can be at home with your kids or your elderly parents if women do more of the caregiving. And that means that women can take the jobs that are the really higher paying, what I call the greedy jobs in society.
Oh, now you mentioned greedy jobs.
Yeah.
So what is that?
Okay. So greedy jobs, whenever I mention the word, people say, oh yeah, I know about greedy jobs. And then I say, no, I have a definition. And the definition is simply that if you work twice the number of hours, you get more than twice the pay. So it's sort of a non-linear shape to the earnings function. But it could also mean that it's not just the number of hours, but the fact that you're called to work late at night, you're expected to work on weekends, you're expected to work on your vacations. That is clearly what we mean by sort of greedy, but the clear definition is that wages are not linear with respect to hours, but they're non-linear, they're convex.
I would think that was the employer that was greedy, but you didn't understand. But,
where there is a job, there is an employer.
Yeah. So, but I mean, is it the employee or the employer that is greedy?
People who are self-employed could impose it on themselves as well.
Yeah.
And of course, it depends upon what the market, what the clients are willing to give. So it's one big, great economic system.
Thank you. I have heard that you have called your students your muses, so I'm very interested in knowing what you meant by that.
Well, what I mean is that we all create knowledge, and for some of us, when we create knowledge, if we don't talk about it, we don't know whether it has real meaning. And so I always tell my students, it's very easy to write down words, but when you speak the words to someone and you look at them, you can see whether they really understand what you're saying. And until I speak it, I'm not quite certain whether I really know it. And of course, they're my muses because they talk back to me. Sometimes.
So what is a recent idea that you have got from from one of your students?
Oh, I can't say a recent idea. Well, yeah. I mean, these can be complicated. I don't want to bore the audience, but I was telling them about applying something called the Coase theorem to divorce. Whether, in fact, if you have two regimes, a regime in which you need mutual consent or a regime in which you can have one individual just walk out, whether these two regimes would lead to a different outcome if you have what's called transferable utility. You don't have to understand any of that. Okay. So the answer that I was trying to convey to them was that there would be no difference if you have a transferable utility under most circumstances in which you didn't have transactions costs. And one of my students said, but in this paper we read, it said that this was bigger than that or something, and then I had to think hard. Okay. Yeah. And what I did was I said, well, we'll talk about that next time. So what I do sometimes, just like with food, that there are leftovers, I'll begin the class with no one likes leftovers, right? And so I begin the class and I say, we really have to have these leftovers. Yeah.
Thank you. And now it's time for our leftovers. Questions from the audience, what I have missed. So now this is your opportunity to fill in the gaps. So who wants to go first? We have a microphone over there. And I also want to say, please be brief. One sentence and a question mark. That's a good question. One question in the front here.
For Professors Weissman and Karikó, I was curious if you could talk a little bit about the development of the delivery vehicles for mRNA and how that convergence research came about.
So I do a lot of work now in lipid nanoparticles. I'm kind of crossing over into your guys' territory. I apologize for that. But when Katie and I started working with RNA, we recognized the difficulty of how do you give it to a person? Because just about every fluid and cell in our body has enzymes that degrade RNA. So we studied maybe 50 plus different formulations, trying to find what worked best for delivering RNA. And after Katie left, we had both tried for 10 years to get lipid nanoparticles. We finally got them, and they were fantastic for delivering RNA and became the basis of the vaccine platform. We later found out that they had a special property known as an adjuvant activity. So adjuvants stimulate the immune system and are required for a vaccine to work. That was unknown and unexpected, but it's the combination of both the RNA and the LNP are critical to make the vaccine work.
But you know, there are other cases when you don't want to activate the immune system. We are using different kind of particles. There are also polymers and other, not just lipid-based delivery.
Thank you. Next question. The gentleman in the middle here.
I'm wondering what is the most exciting development in the next 5 years for quantum dots, the things that get your juices going that you think will make a real difference in either engineering or medicine?
Do you want to take that, or should I take it? Okay, sure. You know, when I go to conferences, the communities become quite large, and there's all these really exciting talks going on, and there are two areas that I think are interesting to me. And I don't know, 5 years is still a long time. I don't know what will come out, right? You see all these beautiful things that are really at the fundamental level. And one of the things is using the surface reactions to do photocatalysis, for instance, to make new molecules or to use them in the way that Louis started way back in the 1980s, looking at the transfer of electrons from the particles to molecules on their surface. That's still an area of quite a bit of interest. The other area is something that the field has discovered, and we're working on it also, is the connection between the kind of light that can come out of single particles and quantum information. There's a big push to create new materials that will create special quantum light, let's say, that can be used in communication or quantum computing. And there's the possibilities that some of the materials that we work on could provide this kind of special light. We'll know in 5 years, I think, is a good time scale to know that.
Please wait for the microphone. Thank you.
Hi, for Dr. Brus. You're a member of the Sputnik generation that was spoken about, that came up after the war, and that sort of drive for innovation that really led America to scientific greatness. Could you do you still see that today? As you look back and see where we are today, do you still see the U.S. at the forefront of science, or is that changing now, in your view?
No, I don't think it's changing. The U.S. enterprise remains very strong, and there are people all over the place inventing things that we don't really anticipate. And when a university recruits new faculty, they recruit from all over the world, assistant professors and senior professors, and they're still coming. Professors are still coming from all over the world. And the technologies which have transformed our lives, semiconductor technology, the software, the applications of the software, what Bill Gates has done, and all of that, those are primarily American inventions, so I don't see any evidence that things are changing.
Thank you. Question over here.
So congratulations, you now have this great title, and you're a laureate, but what actually is there something else that you'd actually like to be known about, that you're excited about, or that you want people to know about you, or that you actually would like to talk about?
I always like to brag, and that's what actually when we met at the machine also that I bragged about that I can make RNA, because Drew is quieter, and I brag about, when I present usually that my daughter is a 2-time Olympic champion, and and I used to be Susan's mom when she was inducted Hall of Fame and different events.
Yeah, I have just a question for you. What advice would you give to a young scientist who is going to start his PhD in a few months, and especially in solid-state chemistry?
Okay, well, I'll take that question, since it has to do with solid-state chemistry. I think the most important part is and this was mentioned before, to be curious. I think curiosity is really the secret to success in basic science and also in applied science, because you encounter a lot of problems and you want to know why you have these problems and what are the solutions. And I think it's like curiosity and hard work. I think that's much more important than smartness in some ways.
I mean, the key thing is to find a unique problem. That's harder than solving the problem, actually, in all honesty. And if you find your unique problem, you have to work it out slowly over time. You don't have to be a genius to work it out, but you just have to be interested. And the more unique the problem is, the harder it will be to convince your colleagues that it's interesting, because they have no experience in this and so forth. So it's a little bit of working in the wilderness for the time being, something like that.
Thank you. In the front here.
I have a question for Professor Golden. You've studied technology change in the past, and we know that technology change is coming to today's labor market and tomorrow's labor market. So from the standpoint of gender equality, what are you looking out for in the technological change we're expecting in the years to come?
Yeah. I'll address that a bit. So the first thing I want to say is that I'm an economic historian, and I do really well predicting the past. I'm terrible about predicting the future. I can give you lots and lots of examples where someone will show me some new technology and I'll say, what's that good for? You know, cell phone. Yeah. However, I think that this is in the realm of what we were just talking about, that we always had the ability to conference with others across the world. We always had the ability to work at home and work remotely, and we didn't coordinate on that. So sometimes it isn't the technology. Sometimes it's the institution, and sometimes it's just the ability to coordinate on it. That's a very big deal. That's where governments and public policy sort of fit in. Another technology that I worked on is the birth control pill. The birth control pill was approved by the FDA in 1960, but young women who were not married couldn't get it because of a set of laws, not just in the US and every state, but in many different countries. So it's not just technology. It's also we are surrounded by institutions and we coordinate among each other. Thanks.
Thank you so much. And with that, I'm afraid that we have to wrap up this Q&A. But I think this was also a message for policymakers here, that to put the right conditions in place for research. And coming back to what Marcia McNutt said in the beginning, that we have some food for thought here for the policymakers. So thank you so much, everyone, for attending this seminar. Thank you, Dr. Anna Sjöström Douagi, for your presentation, and thank you, Dr. Marcia McNutt, for yours. We really appreciate it and learned a lot. And thank you to all our 5 laureates. I learned so much from you. I hope the audience also did that, and that we will leave here a bit more enlightened about your discoveries. So thank you, and good luck in Stockholm.
Thank you.